Hi. Next, the example. First example is glycogen storage disorders. The first type or first example is von Gierke disease. We have type 1A and type 1B, but type 1A von Gierke disease is very important, 5 mark shorts, right? The name is von Gierke disease, that is type 1A. It is autosomal recessive. All are very important MCQ point. What is the defect in von Gierke disease? Accumulation of both normal and abnormal glycogen. Mostly abnormal glycogen. Sometimes normal glycogen also, without metabolism, it will get accumulated in liver, kidney and intestine. That is why they will go for hepatomegaly, renomegaly, intestinal accumulation. This enzyme name is very important. Defect of von Gierke disease is glucose 6 phosphatase. Defect of von Gierke disease is glucose 6 phosphatase. If it is present in one organ, means it will convert the glucose 6 phosphate into glucose. Okay. So glucose 6 phosphatase is the enzyme name. Now we are going to see the clinical manifestations because of this deficiency of this enzyme. Glucose 6 phosphatase is not there, means what are all the problems the patient is having, right? Now, clinical manifestations. One is hypoglycemia, fasting hypoglycemia. This is otherwise known as, because the whole night the glucose 6 phosphate is not able to convert into glucose. The patient is on hypoglycemia throughout the fast. Second one is lactic acidosis. I will tell you the explanation or reason for these causes. First, you should know about the name of the clinical manifestation. Second is lactic acidosis. Third is hyperlipidemia and also hypertriglyceridemia. Both triglyceridemia or hyperlipidemia. Fourth one is hyperuricemia. How the uric acid will get accumulated in this von Gierke disease. Hyperuricemia, one of the important manifestation for von Gierke disease. The last one is megaly, hepatomegaly and renomegaly. So hepatomegaly leads to cirrhosis and renomegaly leads to disturbances in the absorption of the compounds. So these are all the clinical manifestations of von Gierke disease. Now we will see one by one. Hypoglycemia due to the defect of glucose 6 phosphatase, right? The problem is glucose 6 phosphate is not able to convert it into glucose, okay, in liver. Second one is this leads to accumulation of glucose phosphate, 6 phosphate in the blood. So this glucose 6 phosphate undergoes glycolysis in muscles, produces finally pyruvate. Glycolysis means end product is pyruvate. In this situation, this pyruvate is converted into lactate, because in the muscle it occurs, it happens in the muscle. So this pyruvate is converted into lactate. This lactate is usual situations. What will happen is enters into the liver and they will go for gluconeogenesis. Here the problem with the help of lactate dehydrogenase, which is present in the liver. Here the problem is it will be present in the muscle and also blood. So they will get going on accumulated in blood and muscle, and lactic acid production will be more, means lactic acidosis. So first point is hypoglycemia, second one is lactic acidosis. Third point, how this lipidemia and triglyceridemia. Because of this accumulated glucose 6 phosphate, glycolysis is increased whereas gluconeogenesis is decreased. You got this point? Glucose 6 phosphate is available enormously. So this glucose 6 phosphate enters into glycolysis. So glycolysis will be stimulated. Simultaneously gluconeogenesis is decreased, right? So leads to increased production of NADH, NADPH, glycerol and acetyl-CoA. So from the glycerol, TAG synthesis will be increased. From the excess of acetyl-CoA, ketone bodies is increased. From the NADPH, fatty genesis is increased. Like that, all the lipid compounds will be increased in this disease. That is why hypertriglyceridemia or hyperlipidemia. Okay. The next one is hyperuricemia. This excess glucose 6-phosphate diverted into HMP shunt pathway. In this shunt pathway, ribose sugar enters into the purine metabolism, which will produce uric acid. The end product of purine metabolism is uric acid. So excess of uric acid will go for gout disease, hyperuricemia. So hyperuricemia means excess glucose 6-phosphate enters into pentose pathway, that is HMP shunt pathway, where this glucose 6-phosphate is converted into purine nucleotides and finally produces uric acid. So excess of glucose 6-phosphate means excess of uric acid, leads to hyperuricemia. The last one is deposition of both normal and abnormal glycogen in liver and kidney produces hepatomegaly and renal. Okay, so these are all the clinical manifestations. Finally, two important points. How will you investigate this case? Enzyme study in blood. You have to measure all the enzymes in blood. Second one is go for genetic study. These two are the laboratory investigations for von Gierke disease. And how will you manage nocturnal gastric infusion, that is continuous infusion of glucose for the whole night, 12 hours of the whole night.